Hi, everyone, and welcome to this webinar, Sequencing Analysis Viewer, or SAV, a Beginner's Guide. I first want to mention that this webinar is geared toward beginners, so I'll be focusing on the basics of SAV and not in-depth troubleshooting. The information in this webinar can be applied to all Illumina's platforms and will not focus on a specific instrument. For more in-depth troubleshooting for a specific instrument, please see Illumina's support site for more advanced webinars. With that said, let's go ahead and get started. Our goals for today are how to learn to load data in SAV, learn what information is available in each tab, and enable run troubleshooting through an understanding of SAV data. Let's first cover what SAV is. Sequencing Analysis Viewer is an application that gives you the capability to view important quality metrics that are generated from an Illumina sequencing run. One thing to note is that SAV is simply a viewing software so that you can view your run metrics during or after a run. SAV does not perform analysis nor manipulate data. Here you can see the main screen in SAV for each instrument. The overall look and feel of SAV is the same across all platforms. Though the general layout is the same, some metrics will be customized differently across the various platforms and will be displayed as such in SAV. For example, the iSeq 100 has 1 lane and only 16 imaging tiles, while the NovaSeq has up to 4 lanes with more than 3,000 tiles. So you may see many more colored tiles on the left side of the screen in SAV for the NovaSeq compared to the iSeq 100. For this webinar, I will discuss the following items in detail. First, I will go over loading a run in SAV and then discuss each tab shown in SAV, which are the analysis, imaging, summary, and indexing tabs. Throughout the webinar, I'll also point out areas that will help when troubleshooting runs. First, there are certain files required to properly open a run in SAV. Let's go over how to load and open a run. We first need to download SAV, which can be downloaded onto the instrument or onto your personal computer. SAV is a free software that can be downloaded from the Illumina website through the link here, and SAV can be used to view runs from all instruments. First, make sure your computer meets the requirements needed for SAV. The information presented on this slide is also outlined in the release notes on the website. The majority of all install errors are caused by incorrect computing requirements, so please check these out prior to installing. Of course, if you do encounter any errors when installing SAV, please call technical support for help. One thing to note is that SAV is not compatible with Macs nor Linux-based systems, so you will need to use a Windows-based computer when installing SAV on your personal computer. The latest version of SAV is version 2.4.7. I recommend using the most current version of SAV that is available on the website when installing on your personal computer. When viewing runs on all instruments with the exception of the MySeq, I also recommend using the most recent version of SAV. It's when viewing runs on the MySeq, though, you'll want to use version 1.8 due to the computing requirements of new versions. MySeq runs can be viewed on your personal computer using the most recent version of SAV. Generally, each new version introduces a new feature. For example, the version 2.4.7 added percent occupancy and percent pass filter information to the analysis tab, which was not available in the previous versions. Also note that all versions retain the same core metrics. When loading a run in SAV, the files required are run info, run parameters, and the entire interop folder. The thumbnail images folder is not required, but having the thumbnail images present in SAV can be useful for troubleshooting. Note that the thumbnail images are very large files, so having these files available may not always be an option, and it may be beneficial to only have a subset of thumbnail images for certain cycles rather than the entire run. The image on the right shows the different files and folders within the entire run folder, and all the files needed for SAV can be found in this run folder on the instrument, in the run folder in the designated output location, or on BaseSpace. One important note is that you'll need to have all these files in one folder so that SAV can load the run. If you are running SAV on the instrument, you can browse directly to the run folder or to the network location. The MySeq temp folder is shown here in this example. If you're on a personal computer, you can browse to the run, which may be on the computer, a USB, or the network. Here you can see the interop folder, the thumbnail images folder, the run info file, and the run parameters file within the run folder. Rather than browsing for the run folder, you can also copy and paste the file path into the search, then click refresh, which will populate the data from the run listed. You can also view your BaseSpace runs in SAV. If your run was sent to BaseSpace and you would like to download the run files to view in SAV, the steps to download will be the same regardless of what BaseSpace view, classic or new, you are using. From the run page, select the file icon, hover over download, then select run. Ensure SAV is selected and download. Once the download completes, you can unzip the file and view the run in SAV. Next, let's discuss what is reported in the Analysis tab of SAV, as this will be the first tab that populates when you load your run in this software. The Analysis tab contains the majority of information at a glance, so we will focus on this tab for the majority of the webinar. Before I dive into the Analysis tab individually, I want to go over a few tips for interacting with SAV. The chevron in the upper right corner of each graph will expand the graph to the full screen so that you can view a larger version. The axes will auto-scale for each run depending on the values in the X and Y axes. You can fix the scale, which may help you visually compare multiple runs to each other. For example, I can fix the intensity scale between 0 and 20,000 so that I can see how the intensity values in this run look compared to another run. You can pan or move an image by clicking and dragging with your mouse. You can use your mouse wheel to zoom in and out so that you can see a specific area. Note that the values will auto-scale when you're zooming. To zoom in and focus on a specific axis without auto-scaling the other axis, you can use the mouse wheel directly over the axis. And if you right-click on the original image, you can auto-scale the axes, copy the image to the clipboard, or manually scale the axes. Now let's go to the Analysis tab itself. In this image, we have a high-seq run as shown by the 8 lanes of data visualized in the flow cell chart on the left. You can also see the data by cycle, QScore distribution, data by lane, and QScore heat maps graphs on this tab. Let's start by looking at the QScore distribution graph in the top right corner, since this gives us a very general overview of the yield and quality of the run. This run has 484 gigabases yield and 90.4% Q30. Yield and Q30 scores are important because this is what Illumina uses to determine whether or not a run meets Illumina specifications. Yield, which is also referred to as output, is the quantity of data passing filter, and Q30 is the quality of data produced by a run. Q30 is the probability of an error in base calling, so high Q30 means low error in base calling or high quality sequencing data. By the way, if you're interested in learning how much space sequencing runs can take up in terms of mega or gigabytes, for example, check out the approximate sizes bulletin linked at the bottom here. Q30 comes from a Q-score, which is based on the Fred scale. A Q-score is the chance that the base call is incorrect, and the instrument's control software calculates Q-scores after cycle 25 during a sequencing run. A Q-score greater than Q30 means there is greater than 99.9% accuracy that the base call is correct. Illumina's Q30 specification is average across the entire run and is not on the per cycle or per read basis. The yield and Q30 specifications vary for each kit and each instrument. The specifications for a specific run can be found on the Illumina website. Go to Products, Instruments, Sequencing Platforms, and you will see all the sequencing platforms, including the benchtop sequencers, iSeq 100, MiniSeq, MySeq, and NetSeq, as well as the production sales sequencers, HiSeq and NovaSeq. Select your instrument. Here, MiniSeq is selected. You'll be then taken to the MiniSeq system page, where you can select the specifications from the secondary menu to be brought to the specs page itself. On the specifications page, the kit type is in the gray header. For the MiniSeq, these are high output, rapid, and mid output kits. For the different read lengths for each kit type, the total time and output or yield is reported. Further down the page, the expected reads passing filter for each kit type is also listed, but please note that the yield takes into consideration the reads passing filter, so yield and Q30 are the specifications used to assess if a run is above Illumina specifications. Lastly, the Q30 metrics are available toward the bottom of the page and are dependent on the read length. For example, a 1x75 cycle run has a greater than 85% Q30 specification, while a 2x150 cycle run has a greater than 80% Q30 specification. Before we get back to the SAV analysis tab, I want to go over pass filter, because only clusters that pass filter are included in the yield and Q30 specifications. Imagine these are our clusters, and remember each cluster is composed of copies of the same DNA sequence. In this image, we can easily distinguish between clusters, and they're easy to tell apart. Actual images look more like this though, where the clusters are much closer together, which makes it more difficult to distinguish each cluster. In a normal run, if there are two clusters with the same signal in cycle 1, the instrument software may not be able to distinguish the two clusters and will call them as one cluster. The software will keep looking at multiple cycles though. So in cycle 2, if the signal is different between the two clusters, the software will be able to distinguish the two clusters even if they go back to the same signal in cycle 3. If the run has a low diversity library or is overclustered though, the bases may be the same for each cycle, and the software may mistakenly call these two clusters as one cluster. This one cluster would then not pass filter, and the data for both clusters would not be used in the run. Note that for pattern flow cells, since the location of the clusters is fixed, empty wells or wells with polyclonal clusters will not pass filter. Please see the technical note here for more details on passing filter, in addition to the bulletin on why sequencing 25 or more cycles in read 1 is recommended. Now that we've gone over yield, Q30, and pass filter, let's go back to the Analysis tab in SAV and break down the QScore distribution plot in more detail. The QScore distribution shows the number of total reads versus the QScore. The QScores reported include the quality score of every cycle, and only reads that pass filter have their QScore reported. Illumina bins the scores, and anything above Q30 is graphed in the green to the right of the green vertical line. Here you can see some small blue bars with Q-scores below Q30, and you can also see green bars above Q30, where most of the data is. The yield and Q30 are reported here, so you can easily check if your run meets Illumina specifications. This plot has the following drop-down options. In lane, you can view the Q-scores for each individual lane or all lanes in your run. In surface, you can view the top, bottom, or both surfaces. In read, you can view read 1, 2, 3, or 4 if you'd like to look at a specific sequencing or index reads. In cycle, you can view the Q-scores for any given cycle or all cycles in the run. The plots are automatically scaled, but you can fix the scale by checking this box. And similarly to what I pointed out earlier, the chevron on the top right will expand the graph to the full view of the screen or minimize the graph back to the pane view. Next, let's look at the data by cycle plot. If a run is not meeting Illumina specifications, this is a great plot to investigate what is causing the poor performance. In data by cycle, you can follow the progression of metrics during a run to determine if anything is abnormal. You can select the metric such as intensity, Q30, or percent base. You can break down the graph to look at specific lanes, surfaces, or channels through the drop-down list. The plots are auto-scaled by default or can be fixed by checking the Fix Scale checkbox. For some metrics, such as Q30, you can monitor the metrics for the entire run up to that cycle by checking the accumulated checkbox. Here's an example of a plot looking at the metric intensity, all lanes, both surfaces for channel C. Let's go over some of the data by cycle metrics in more detail, and here are the options you can view. When intensity is selected, the intensity by color is shown. Since 4-channel chemistry uses a mixture of nucleotides labeled with 4 different fluorescent dyes, runs using 4-channel chemistry will show the intensity values in the 4 different channels as shown here. Since 2-channel chemistry uses only 2 different fluorescent dyes, runs using 2-channel chemistry will show the intensity values in the 2 channels. Note that the new NextSeq 1000 2000 platforms utilize blue-green chemistry as opposed to the red-green chemistry. 1-channel chemistry uses only 1 channel for imaging, but there are 2 images taken due to the chemistry step, so we still have 2 channels for the intensity values. Please see the technical notes here for more details on the different chemistry types if you're interested. Next, let's go over the quality graphs and data by cycle, which will be generated after the 25th cycle. And let's focus on the % greater than or equal to Q30 graph. A high quality run will have Q30 values throughout the run, and it's normal for the Q30 values to decrease as the number of cycles increases, as you can see the slight negative slope here. The blue dots correspond with the individual tiles that are outside the standard deviation for that cycle, and these can be useful to look at when troubleshooting bubbles or similar issues that may cause changes from cycle to cycle. There's also the median QScore graph, which graphs each tile over all bases. Next is the full width half max metric in the data by cycle graph. Full width half max defines the proportion of a cluster used to detect signal. The instrument looks for pixels that are close to the brightest, most central part of the cluster, which allows for better separation of neighboring clusters. Technically speaking, full width half max is the width of the spectrum curve on the x-axis measured between the points that correspond to half the amplitude on the y-axis. In SAV, full width half max is a good indicator on how well the instrument is focusing on clusters, and you may hear this referred to as the focus metric. If the instrument is focusing well, this graph is ideally flat. If the full width half max is spiky or erratic, there may be focusing or optical issues. Note that a low diversity library can cause the full width half max to have more variation compared to a well-balanced library and will not appear as flat, but an erratic full width half max indicates a focusing issue. Let's take a look at the % base graph. This graph reports the percentage of clusters that have been called for a given base, and specifically, the graph will break down what bases are being called in a cycle. In this run, the library is well balanced with roughly 25% of each bases being represented. Here is a low diversity library, and you can see the bases being called up to almost 100%. This plot can even be read as a Sanger plot because of how individual bases can be called for each cycle. This type of low diversity library needs PHYX or other balanced libraries to help balance the signal in order to achieve high quality data. For example, here's an amplicon run pooled with many other amplicons. So even though each amplicon individually has low diversity, when pooled together, the base diversity is balanced between about 20 to 30%. For more information on low diversity libraries and how they can affect your sequencing, please see our webinar on low diversity libraries. The phasing and prephasing values are used to determine how good the chemistry was. Phasing is when molecules in the clusters are behind a cycle, and prephasing is when molecules in the cluster are ahead of cycle. There are 2 ways to visualize phasing and prephasing. In the majority of clusters, the C base is being called. In phasing, the base G from the previous cycle is being called, and in prephasing, the base A from the following cycle is being called. Illumina does not have specifications on phasing and prephasing, and these values are used as a secondary metric in conjunction with another metric. For example, if phasing and prephasing values were higher than normal and the Q30 was low, the phasing and prephasing could help pinpoint the cause of the poor quality. The data by cycle pane also plots the corrected and called intensity on some instruments. We're not going to go into too much detail on these graphs because they are not commonly used in troubleshooting, but please refer to these slides as a reference if needed in the future. Briefly, the corrected intensity corrects for crosstalk, and the called intensity shows the average intensity for all clusters. Here are a few additional metrics in the data by cycle graph. In the percent no calls graph, the amount of N, or no calls per cycle, is shown. The error rate will be populated if phi x is spiked in a line because the error rate is based on the error of the known phi x sequence. If phi x is not aligned, this plot will not populate. Note that having the error rate for phi x helps with troubleshooting, so we always recommend spiking in phi x. Even 1% for a well-balanced library can be very helpful. The signal-to-noise graph, which plots the signal to background in the cluster images, can give us information on the quality of the clusters or the imaging of the clusters. These values are generally between 8 to 12. Let's go back to the Analysis tab, then go over the flow cell chart on the far left. I find the flow cell chart very useful because it breaks down the specific values in each tile on the flow cell, while the other graphs are an average for the metrics. There are 20 metrics that can be plotted on the flow cell chart as shown here. The flow cell chart can be broken into top and bottom surfaces, a particular cycle number, or a specific channel. I often like to pair the data by cycle graph with the flow cell chart so I can see how the individual tiles are affecting the overall metrics, such as the intensity plot in the data by cycle graph, compared to the intensity of the individual tiles in the flow cell chart. Here are some examples of the options you can choose in the flow cell chart. This chart shows the intensity, all services, cycle 1 for channel A. I can also look at the Q30 for the bottom surface of cycle 96. Both graphs are showing a high-seq run with 8 lanes. Here is the Q30 for all surfaces in cycle 14. If you hover your mouse over a specific tile, the lane, tile number, and value will be reported. One thing to note about the flow cell chart is that the scale on the right is auto-scaled. Sometimes the colors can be alarming and that there could appear to be large differences between the tiles, but the difference in scaling may actually be very small. Like here, for example, we see the Q30 values are between 95.4 and 97%. So the quality values in the blue tiles are actually very similar to the quality values in the orange tiles. Note that you can fix the scale and adjust the axes if that helps visualize. Here are the density and density pass filter graphs in the flow cell chart. Note that here, the libraries are similar across all lanes, but each lane could have different density and pass filter values depending on the libraries. These graphs are useful for troubleshooting if a run is overclustered, and a run with normal cluster density will have an expected gradient as shown here. Due to the fluidic system, the cluster density is highest at the inlet, which is visualized at the bottom of the flow cell chart for the high-seq graph here. The cluster density value should not vary significantly between the lanes and tiles. With an overclustered run, you may observe that the cluster density is highest at the outlet, and you may see tile-to-tile variation rather than the nice gradient. If we go back to the Analysis tab again, we can review the data by lane graph. For this plot, there are a few metrics that we can view, including density, percent occupied, and percent pass filter. The different surfaces can also be viewed in this graph. Note that percent occupied will only be shown for iSeq 100 and NovaSeq runs, and this metric can be very useful in determining if the run is optimally loaded or not. If density is selected in the data by lane graph, The blue box plot shows the raw cluster density. If the run is using a pattern flow cell, the raw cluster density will be the same for all lanes due to the fixed number of wells. The green box plot shows the clusters that pass filter, which will be different for each lane for all flow cell types. The red lines represent the median value, and the error bars graph the minimum and maximum values without outliers. The blue numbers above the lanes on the x-axis, which is 96 here, tells us the number of tiles used for analysis, which can also be visualized in the flow cell chart. The last chart in the Analysis tab is the QScore heat map in the bottom right corner. The QScore heat map is a visual representation of the QScore distribution. This graph is a nice visual representation of the quality and could be a good graph to show in presentations. It's not often used for troubleshooting though, so I won't discuss it much here. Similar to the other graphs, the QScore distribution graph can be viewed by lanes and surfaces. Now that we've gone over the Analysis tab with the run graphs, let's take a look at the Imaging tab, which shows the images from the sequencing run. These images are great for troubleshooting, but are not used to evaluate the performance of a run. The Imaging tab shows the thumbnail images in each tile, and these images will only be present if the thumbnail images folder is present in the run folder. These images can be sorted by cycle, lane, surface, read, and tile number. With these options, you can file one tile throughout cycles or look at specific situations such as only the top surface or only an index read. There are also options such as copy, export, sort, create custom plots, and filter. Here are more details about those options. You can select all rows or copy selected rows. The table can be exported or columns can be sorted. You can also create custom plots, which can be useful for instruments using pattern flow cells. You can also choose what columns to display, filter columns, and move up and down when viewing the thumbnail images. Here's an example of using the scatter plot function. You can plot the tile versus density in a plot with tiles on the x-axis and density on the y-axis will appear in a separate window. You can then compare the flow cell chart to this plot, which shows the cluster density is highest at the inlet tiles compared to the outlet tiles. Here is a list of the metrics available for the plot. One common graph is the percent occupied versus percent pass filter graph. We often use this to assess flow cell loading for NovaSeq and iSeq 100 runs. Technical support authored the bulletin linked at the bottom of the slide to provide details on such plot if you'd like to take a look at that. Here's an example of clicking on the flow cell in the flow cell chart and how that tile's image will open in the Imaging tab. Note that this is only available if the thumbnail images are present and saved in the correct file pathway on the run. Click the tile, which will open the image in the right. It will open the image in the Imaging tab. If you click on the bases A, C, G, and T in the top right, this will toggle through the different channels in that tile. Next is the Summary tab that contains detailed numerical metrics for the run. The Summary tab shows the numerical hard values of the graphs observed in the other tabs. The sections are broken down into cycle status, run summary, and each read in the run, reads 1, 2, 3, and 4 if present. The cycle status shows the cycles that have been extracted, called, and scored, which should be the total number of cycles in the run. The error rated is the cycles that have been aerated using phi x. So if no phi x is aligned, this value will be 0 as shown here. In the run summary, the yield, projected yield, percent phi x align, percent phi x error rate, intensity values at cycle 1, and the Q30 are shown for the reads. Each read will have this information broken down with the metrics specific for each lane. Here is a high-seq run with 8 lanes of data. The cluster density, pass filter, and phasing, prephasing metrics are also shown here, as well as the Q30, yield, percent phi x aligned, error rate, and intensity. Finally, let's move to the indexing tab. The indexing tab is only available if the sample sheet was supplied at the start of the run, because the indexing tab pulls the index sequences from the sample sheet. Here you can see there are more than 50 samples, as shown in the table on the left and the graph on the right. The indexing tab shows the percent reads identified, which is the fraction of passing filter reads assigned to an index. Please note that phi x does not have an index, so if any phi x is aligned in the run, the percent of phi x aligned will decrease the percent of reads identified. For example, if you have 10% phi x aligning, the maximum number of reads identified would be 90%. The indexing tab also breaks down the percentage of reads identified for each sample, so you're able to see how each sample in your pool is distributed. Before I finish this webinar, I'd like to point out some great resources and links. The SAV support page has links to the SAV software guide, as well as the downloaders for the different versions of SAV. For support regarding all Illumina topics, please visit our technical support bulletins page and training videos in the links provided. Again, I'd like to highlight the technical bulletins page. Nearly all of the bulletins you'll find here have been authored by technical application scientists like myself, since we're aware of the common questions and issues that customers have. I'd highly recommend checking these out, and if you're looking for a specific one, just please ensure you scroll to the bottom of the page so that they're all present. And again, the training page. On this page, one can find training videos, video tips, recorded webinars, as well as upcoming webinars. Thanks so much for your time today. Should you have any questions, please add them to the chat. Thank you.